Bismillahirrahmanirrahim. Dear students, hope you are fine. Just to recall you that we were discussing about the isomerism and we had discussed about the types of isomers. That is, we have seen that the isomerism could be of two main types. One of them is the structural isomerism and second one is the stereoisomerism. Today we will focus on stereoisomerism. The term stereo arises from the Greek stereos, which means solid, and is used with reference to hardness, solidity, or the three-dimensionality character of the matter. The stereochemistry is actually the branch of chemistry which deals with the study of structure of chemical compounds in three dimensions. Or we discuss the spatial arrangement of atoms in a compound. Dear students, it is quite clear that 3D study of the molecules is very important in understanding the all characters which a compound shows. But mostly in our daily life, we are dealing with the screen or a paper at which we have to draw the structure of a molecule in 2D representation. So we use different type of signs which we will discuss soon. But let first understand what the stereoisomerism is and what is the importance of stereochemistry. So there are many compounds which have the same formula, same sequence of atoms, but they are quite different from each other when we observe them. For example, while examining the cis-2-butene and trans-2-butene, we discover that in both of the isomers there is a great difference between their characters. Let's suppose discuss about the melting point and boiling point of these two compounds. It has been observed that cis-2-butene has melting point minus 139 degrees Celsius, while the trans isomer has the melting point minus 106 degrees Celsius, while the boiling point of both isomers is also different. Cis-2-butene shows 4 degrees Celsius boiling point, while the trans-2-butene shows 1 degree Celsius boiling point. And we expect the difference in their physical characters because we see that all the atoms in both of the isomers are arranged in different sequence. You can see the two-dimensional structure of both molecules and the three-dimensional structures on your screen. And you observe that in both of the cases, the sequence of the atoms, although the connectivity is same, sequence is same, but their angles to which they are attached is different in both of the cases. This results difference in the electrostatic attraction. For example, see this hydrogen and its environment. The hydrogen at the last carbon and first carbon, they are close to each other, though so they might expect to have different influence on each other due to their electrostatic charges. While in case of the trans isomer, you see that the hydrogens on the one carbon number 1 and hydrogen at the last carbon, they are farthest from each other, and they should have different electrostatic forces of attraction or repulsion. That is why their physical characters are different in both of the cases. This is also seen in the 2D structure of the molecule. You see that both of the methyl groups are at one side of the plane, and in trans-2-butene they are at trans position or across the molecule. So the electrostatic forces which are developed around an atom are quite different in both of the cases, which results in the difference in their physical characters. And you see that their melting point and boiling point are quite different. Moreover, in many cases, there is also a difference in their chemical characters. For example, here is an example of cis-butene dioic acid and its trans isomer, the trans-butene dioic acid. The cis-butene dioic acid readily eliminates water on heating, giving a cyclic anhydride. You see in their structure that in the cis isomer, both the hydroxyl group are at the same position, so the water is readily eliminated from their molecule, giving the anhydride. And here is the reaction. When water is eliminated by heating from cis-butene dioic acid, the cyclic anhydride is formed, while in case of the trans isomer, both of the hydroxyl group are situated furthest, and it is quite hard for this molecule in the trans form to eliminate water and to gain such a cyclic structure. As well as their physical characters are also different. For example, the cis-butene dioic acid has the melting point 142 degrees Celsius, while the trans-butene dioic acid has the melting point 300 degrees centigrade, which is quite a large difference. Dear students, mostly the biomolecules which we see in our life are stereospecific. This means that mostly they have a stereoisomer which has a quite different properties from its other counterpart. Mostly the enzymes, amino acids and drugs which are of bioorigin compound, they are stereoselective, and their stereoisomer has no or sometimes totally different behavior when they are taken. You will see many of the examples in the next coming lectures. Here is an example of S and R thalidomide, which is a very famous compound and has been used largely as a sedative for the treatment of pregnant mothers to make them calm. In the initial instances, when the when the scientists were not able to see the stereochemistry and they were given the drug to the patients for the sedative nature or sedativity character of the compounds, but they observed that this also caused some dysfunction in the fetus of the mother. This they observed in the early 60s of 19th century. And from that era, the exclusive study of the stereochemistry or the three-dimensional structure of the molecules begin. Later on investigation, they found that the thalidomide has two types of isomers. One of them is the R isomer and second one is the S uh, isomer. We will discuss the S and R types of isomers very soon in the coming lectures. But now just concentrate on the these drugs and their action. They found that the S thalidomide has a sedative action while the R thalidomide has teratogen or it causes the birth defects in the fetus. Although while you see the structure of both of the compound, there is a slight difference between them. You see that here the hydrogen is at backward position while this whole molecule is at in front of viewer coming out of the plane of paper, while in case of its R counterpart this case is different. The portion of nitrogen bearing structure is backward in position while the hydrogen is in front of coming out of the plane of paper, which is denoted by this wedge in their representation. This is slight difference make the properties of the compound totally different. One of them is a sedative in action while the second one causes defects in the fetus. So it tells us that the stereochemistry is very important very important to study, especially in the bioorigin molecules. Many of the drugs are having such a character. Similarly, you may have heard about the ephedrine, which has two isomers which are shown in the screen. One of them is the minus ephedrine and second is the plus ephedrine. They have different structures. For example, in two-dimensional diagram, you see that the hydroxyl group and nitrogen bearing side of both lies at the left position, while in case of positive ephedrine, the hydroxyl group and nitrogen bearing chain both are at right side. You can also see this in their three-dimensional character. The phenyl ring in both of the cases is at backward position. OH and nitrogen bearing chain is at left side, while in case of plus ephedrine, both hydroxyl group and the nitrogen bearing chain is at right side. This gives different stereostructure to both of the compounds, as a result of which they have different characters. One of them is relaxant and is used as a cough syrup, and second one has no action at all. Now we see why does it happens. Just concentrate on the given structure. You see that there are two structures here shown in this diagram. At above, a molecular structure is there, and at the substrate is shown at downward position. You see that the blue side of the molecules is adjustable at blue side, the green is at green side and red one is the at the red side. While in this case, when you rotate the molecule and their stereochemistry is different, blue will adjust at the same position, but at this side there is a red when you turn the molecule from this side to the other side, and it brings the red to this side and the green to other side, so they are not adjustable at the side, so binding will be weakening and action of the molecule at their active site will be non-feasible. That is why the molecule will not show the desired effect. And thus, the stereochemistry of the compounds is essential to study, and exact structure molecule is needed for a desired action. There are many more examples which we can take. For example, here is a compound with different characters you see in your daily life. One of the compound is present in spearmint oil and second one is in caraway oil. Both have different physiological effects and sensation to us. Although the sequence of the atoms in their molecule is the same, but the stereochemistry in both of the compounds is different. And you see this there in the screen you are being viewing, and in their 2D structures as well. The molecule at left side is the the carbon from spearmint oil, or you know in your daily life as mint, while at the right side it is from the caraway oil, and in local language you call it zira, and is extensively used as a spice in the daily meal. And you observe that both of them have very different characters, not only physiologically, but their taste is also different. Even the smell of both of them is very different. You can compare the structure of both. Here is the carbon which is responsible for their difference in characters. This carbon is called a chiral carbon or chiral center. A chiral center is that center at which four different groups are attached. And you see in the two-dimensional representation of the compound that there is a difference in their structure at this point, which makes their characters very different. You can compare the character in 2D as well as in the 3D forms. And you see that both have different structures in three-dimensional, and as well as you can see very easily to differentiate them at their two-dimensional representation. And you see that a slight difference in their arrangement of the atoms, in spatial arrangement in the 3D representation, makes the characters of the molecule different from each other. Dear students, hoping that you have a good understanding about the stereochemistry and about the importance of the stereochemistry in our life. Thank you.